Yeah. So once again, good afternoon, class. Today we are learning about monoclonal antibodies, DNA diagnostic system, and molecular diagnosis of genetic diseases. But they come under the common umbrella of molecular diagnosis. I was mentioning to the class that if a hospital has a good support for diagnosis, for disease diagnosis, then that hospital will make right judgment to start therapy. If the diagnosis goes wrong, then the therapy will go wrong. And to choose the right diagnostic path, right, one has to be familiar with the advancement and fundamental knowledge. So in this particular class, this particular chapter, it focuses on a term called as antibodies. And antibodies, you must have heard in the beginning of COVID infection, it was in all over the place that whether uh, whether those who are infected with COVID, are they having right antibodies, right? So antibodies is one of the arm of immune response. Antibodies, which are present in our body, it's one of the arm of our immune system. And so it was said that if there is large antibody, it may not be protective. So all these discussions were going on in the early, that's why one of the companies called as Regeneron, they developed monoclonal antibodies to COVID infection. And if you remember that at that point of time, somebody very famous in the country got sick in 2020. He got the monoclonal antibodies from Regeneron. So monoclonal antibodies are a special kind of antibodies, and antibodies are one of the molecules that our immune defense system produces to fight infection. So antibodies could be used for treatment. Antibodies could be used for diagnosis. Both. It has huge implication. Before I go into the detail, I would like to discuss a little bit about antibodies. So what are antibodies, right? So antibodies are essentially, maximally, it's a proteinaceous in nature. And this antibody structure, if you see, this is V-shaped, right? Not exactly, but you can see this is the victory sign for the V within the Y, and this is the long tail of the alphabet Y. So this is the antibody, it's the shape of the antibody. But besides this Y-shaped molecule, there are also two chains. So for sake of simplicity, for understanding, we can say that antibodies are made up from 4 polypeptides. 1, 2, 3, 4 polypeptides. Out of these 4, 2 are heavy because they are longer and 2 are light. So now we can divide this molecule into heavy and light. So in this tetrameric 4 subunit molecule antibodies, 2 are heavy chain and 2 are light chain. Now this cavity, this groove, which is in this portion of the V-shaped, triangular groove, right? Pocket. This is where the antibody binds to the pathogen or virus, bacteria, fungus, allergen, anywhere. So an antibody is a protein molecule that is produced by a special type of lymphocyte called as B lymphocytes, right? Now, when I'm talking to you about antibody, you must connect to the stem cells, where I had talked about multipotent cells, bone marrow, which give rise to RBC, WBC, and one of the cells of WBC is lymphocytes, and one of the lymphocyte is B cells. So the antibodies are produced by B cells. And these antibodies, they go and directly bind to the pathogen. So this is a cartoonish structure. let's look at the more molecular structure of antibody, which is very complicated. Antibody is a very large molecule, right? So as I mentioned to you, there are two light chains and two heavy chains, right? So this bluish green or greenish is the light chain here, and the heavy chain is here. Now, these heavy chain and the light chains, I repeat, these heavy chains and the light chains, they have two regions, a variable region called as VL and CL. So VL means variable light chain, C means constant light chain, VH means variable heavy chain and constant heavy chain. Now, why does it says variable? Why this heavy and light chains are divided into two parts, heavy and variable and constant? So the variable portion of the antibodies, it keeps changing. It keeps changing depending upon the foreign particle that enters into our body. So this variability is important because let's say COVID-19, there are many strains. Right now, Omicron is there, and then some other subtype will come or some other strain will come. So everything will remain the same, but the variable chain will change. It will adapt to fight the new variant, right? And so just I want you to, as we are inching towards finishing the semester, it's very important to integrate your thoughts. So when I said COVID-19, it should appear to you that, okay, this is a virus and we're talking of immune mechanism, means this virus can cause disease. I'm talking of variants, means you should go back to the chapter 2 where we talked about mutations or synthesis of DNA on RNA. The basis for synthesis could be, in majority same. So the beauty of this course is that when you integrate, because that's how you're going to, you can come across, many things as we go forward. And healthcare has become very important in this century, and biology has become, because as I mentioned to you, biology and healthcare will be the most attractive profession in this century. Now it's possible that biology could integrate with maths for artificial intelligence, or with computational science for contact tracing, or uh, with recombinant technology, or DNA synthesis, totally chemistry. So, um, so you need to integrate what you have learned, right? Then only you can understand. And there could be, situations where this knowledge could be helpful. So let's look at um, some of the. So I talked about antibody. Now, this is just a simple graph, right? So when a virus, let's say, infects our body, right? So when a virus infects our body, so virus is new to our body and our body is new to the virus. Both are new to each other. But virus has an upper hand because it can trick our body. It can trick our transcription system. It can trick our translation system. It can go inside the cells through the receptor. Cellular receptor, it can go, because viruses have emerged. One of the hypotheses is that viruses have emerged from taking DNA from our body, and then it mutated. So there is an incubation period where the virus tries to establish itself, right? And then when the virus tries to establish itself in our body, we start to get mild symptoms, right? And and at this phase, when the virus is under incubation period, where it's trying to establish itself and it's throwing up mild symptoms, and we are not sure that we are sick, or we are are okay, or it's seasonal, versus so many things. So after then, when the virus establishes itself, it tries to grow in our body, and then it starts throwing up our immune system. it starts battling the immune system. So virus incubation period, then the replication increases because virus is dominating over the cellular functions. And then when the immune system notices the virus, the first type of antibody that is thrown by our body is IgM. Ig stands for immunoglobulin, globulin, and M is M type. And then the IgG. Between IgG and IgM, IgM is early antibodies and IgG is later antibody. But IgM goes down slowly and it becomes undetectable. IgG is stronger and it stays in our body for a long time. So two major antibodies, IgM and IgG. IgM is early and IgG is later on and strong. Now, which cell produces antibodies? So antibody is produced by a cell which is called as B cells. B cells, the starting point is hematopoietic stem cells. One branch of hematopoietic stem cells becomes pro B cell, then large B cell, small pre B cells. So this is a lineage. You don't need to know the detail. So bottom line is B cells are involved in the secretion of antibodies. And antibodies are one of the primary main molecule that tries to fight infection. So now this B cells could be naive. For example, in human embryos or infant inside mother's body, they are not exposed to pollution as compared to adult persons. So there, the B cells are naive, they are new. Even there could be B cells in our body right now which are naive. They have not been exposed to any bug or pollution or any pathogen. Once a naive B cell antibody gets exposed to a pathogen, it reacts. And a certain subset of B cells form memory B cells. This is the memory B cell which is at the center of vaccine protection, right? So we have, if we have taken mRNA vaccine, we have antibodies, but there are some B cells that will be stored in our lymph node at the memory B cells, so that when we get we get infected by COVID-19, these memory B cell will sprung into action and produce antibody to fight COVID-19. Activated B cells are those B cells which are actively participating in immune defense mechanism. And so remember this plasma cells. Plasma cells are the real cells which secretes antibody. So antibody is secreted in the body, means in the blood system. So let's say if I was infected with COVID-19, if somebody draws my blood and you check for COVID-19 antibody, it should have antibody. My blood should have. More than that, I have been vaccinated thrice, first time, second time, then the booster, then it is expected that I should have antibodies to spike protein of the virus, because against which this mRNA has been made. So these antibodies are in my blood because these plasma cells have secreted antibodies against COVID-19. And there should be also memory B cells in my body, because if I get an infection, then I can rely upon these memory B cells, in hypothetically, that they will come back into action to defend me from COVID infection again. And that's the whole logic for vaccine. Now, anything that is foreign in our body is called as antigen. It could be a pollen, it could be a flu virus, it could be a protein, it could be a foreign organ. It is an antigen. Anything that is foreign, and anything that is foreign has the capacity to react with our immune system, or vice versa. Our body doesn't accept foreign doesn't accept foreign foreign organs or foreign antigen quickly unless we are immunocompromised. So, um, so compromise, right? So this antigen, right? So let's say this blue color rectangle is a antigen, and it has the capacity to elicit immune response. Now this antigen could have various domains against which antibodies can be formed. So there can be 1, 2, 3, 4, 5, 6, 7. I'm telling something very important. Just listen to me carefully. So this is one foreign molecule, the bluish color rectangle, shape. Now it can generate 7 different types of antibodies because this is a complex molecule. Antigen is foreign, complex. It can generate 7 different types of antibodies. Now you can ask me, okay, then why doesn't this antibody protect us from this foreign antigen? So it is possible that yes, there are a lot of antibodies, but these antibodies are not protective. Right? So it was seen in early infection of COVID-19 that people threw a lot of antibodies, but they were not protective. So I might have a lot of antibodies when I got infected, but that antibody was not protective. So it's very important that out of these antibodies, some should be protective antibody. Protective antibodies are that antibody that can protect us from infection, that can fight and neutralize the infection agent. I repeat, neutralizing antibody or protective antibodies are those antibodies that can fight the infection and minimize the infection. So that we could have high antibodies to COVID-19, but if those antibodies are not protective, it will be not useful. And the logic for many of the vaccine is that to produce large amount of antibodies that can protect us. Now, B cell is not alone in fighting infection. B cells collaborates with T cells. B and T cells collaborate with neutrophils. They collaborate with other cells. So our immune response is a collective response of various cells and molecules. Immune response or our immunity comes from the teamwork of many cells, T cells, B cells, natural killer cells, neutrophils, many types of cells. So there is a communication within our body. So the immune system is working most of the time is because there's a good communication. If there is no good communication, then so how do these cells communicate? They communicate through antibodies, through growth factors like cytokines, interleukins, and all those things. Now, it has been seen that the cancer cells also express certain kind of antigens, special. So let's say a virus is special because it is different than our body. So it's an antigen. Now, research shows that even cancer cells, they express some of the unique markers that is not present in normal cells. So those are called as cancer antigen. So antigens which are specific to a certain type of cancer cells, they're called as cancer antigen. For example, CA125 is one of the important crucial protein marker for ovarian cancer. Right? In the stem cell when I was teaching, I told that there are 2 CD markers. When they are present, there are high chances that those are leukemic stem cells. Right? So these cancer cells also, they express some unique antigens. But now I am giving you basic information. Now you will see that how these antibodies can be developed into a therapeutic molecule. Right now I'm talking from the basic concept of immunology, B cells. But now I'm trying to little bit switch gear and say that how these antibodies can be utilized to develop a good diagnostic system or good therapeutic, modalities. So first, what I would try to talk something very basic is there are two types of antibodies. One is called as monoclonal antibodies. As the name suggests, mono means one. Clonal means a one cell that can produce one type of antibodies. So a monoclonal antibody is that antibody that can bind to a specific place on a protein or an antigen, right? An antigen could be protein, glycoprotein, lipoprotein. So a monoclonal antibody has the property to bind to only one place on the antigen. A polyclonal antibody is that antibody that can bind to many places on an antigen. So monoclonal recognize only one place and polyclonal recognizes many places. Now, the word I'm using, place, is not the right word. So the place or the portion of the protein or antigen to which an antibody binds is called as an epitope. I repeat, epitope is that region of a molecule that is recognized by an antibody. So a monoclonal recognizes only one epitope, whereas polyclonal recognizes many epitopes. Poly means many, right? So it's a clone of many antibodies that binds to different portion of the antigen. So this is the difference between the polyclonal and monoclonal antibodies. Now, how a monoclonal antibody is generated, right? So which cell produces antibody? Antibody is produced by B cells. And these B cells will produce antibody. These B cells will produce antibody. Now, when I want to make monoclonal antibody, so let's say this is a cell called as B cells. Now, this antibody, we figured out that this antibody is, let's say, is protective to infection by an agent, right? This antibody. So we need more of these antibodies. So what scientists do, they fuse through by means of chemicals. They fuse this B cells that is producing this antibody with a special cell called as myeloma cells. Now, what are myeloma cells? These myeloma cells are cells which are called as plasma cells, but they are cancerous, which means that they can proliferate. Cancer means they are immortal, right? Cancer cells are immortal. So on one hand, you need a B cell that produces specific type of antibody that is protective, but you want this to be abundant. So what is done is that it is fused using a chemical called as polyethylene glycol. So the membrane of both these cells fuses, and when they fuse, they become as one cells. So this is also a type of recombinant cells, hybridoma, two cells. B cells that produces antibodies of interest and fuse to myeloma cells such that it becomes immortal. It can produce this antibody forever. And this antibody can be produced in excess to protect from this infection. And this immortality from, so myeloma cells myeloma is a type of blood cancer. In that blood cancer, there is huge amount of plasma cells. But here, for the sake of technology, you fuse them so that you get immortal B cells that produces antibody in tons. And these antibodies can be used in patients who are having that kind of infection. Right? So that's how the monoclonal antibody is produced. Now, there's a special kind of, so cancer is a heterogeneous disease. It's not a homogeneous disease. It's very complicated. And as we understand more, some cancers are very simple, some cancer cells are very complicated. So there's a special type of breast cancer, which expresses a lot of receptor called as HER2. So these are called as HER2 positive breast cancer. So a normal breast cancer cells, a normal breast cells will produce few receptors, but an breast cancer cell will make a HER2 positive breast cancer cells which will have a lot of HER2 plus receptors. So scientists have developed monoclonal antibodies against this HER2 receptor. So these monoclonal antibodies go and bind to these specific cells, and because of immune mechanism, these cells are destroyed by the immune system. So this is one of the example of immunotherapy. Immunotherapy is that when you use immunological molecule to treat a disease. So here, the immunological molecule is a monoclonal antibody against HER2. HER2 can be overexpressed in certain type of breast cancer. So a patient who will take HER2 monoclonal antibodies, these antibodies will go and bind to HER2 positive breast cancer cells and destroy those HER2 positive breast cancer cells, and the patient can become normal. So what did we discuss until now? We discussed what is an antibody, from where it is made, and its primary role is to defend antibodies, but because of science and scientific development, these B cells can be developed into a monoclonal producing factory, and this monoclonal antibodies can be used for targeting abnormal cells within our body, such as in breast cancer or prostate cancer. So cells, in the body, our cells interact, they talk to each other in many ways. And they try to protect body, but a single failure can cause disaster to our body. This is just a cartoonish diagram how these cells, they work with each other. Okay. Now these monoclonal antibody, I mentioned to you, antibodies are Y-shaped, heavy chain and the light chain. Both these heavy chain and the light chain, they have variable region that changes depending upon the strain of the pathogen. But these antibodies could also be tagged to cytotoxic drugs. So, so let's say, for sake of understanding, as I mentioned to you, that yeah. So this is let's say heavy chain, and this is this is light chain. I'm sorry, this is light chain, and this is heavy chain. Now, within the light chain, there could be variable region, and this is constant region. This is light chain, variable region, constant region. Heavy chain, variable region, constant region. Variable region. So these variable regions, these are variable regions on the light chain and the heavy chains. They change depending upon the type of strain of a particular virus or bacteria. Now, this portion of the antibody, it is very constant. So what scientists are doing, they are tagging this portion of the antibody with a cytotoxic anti-cancer drugs. So this antibody as a whole can be used to target cancer cells. For example, let's say there is a molecule called a cisplatin. Cisplatin can kill prostate cancer cells. But in prostate tissue of humans, it's a gland, and that gland has normal cells and abnormal cells. The problem is you don't want to kill normal cells, you want to kill abnormal cells, which are cancerous. So these cancer cells expresses special antigen, which are called as cancer antigen. So you can design your antibodies, your heavy chain of these antibodies, excuse me, heavy chain, right? Design heavy chain, design light chain against that antigen to prostate cancer. And again, tag this with this special arsenal, cytotoxic drug. So this antibody will go to only those cells which express prostate cancer antigen. And then they'll start an immune mechanism. They will recruit special cells, and then these cells will kill that cancer cells. So this is the advantage of immunotherapy. It can be more specific. Right? So I will just go quickly to the coronavirus. So coronavirus has a protein called a spike protein. This spike protein binds to the ACE2 receptor on the lung cells. And when it binds, the virus goes inside, and it starts from, first the virus will replicate, then breathlessness and asthmatic-like situation, oxygen going down, and then many other complications. But if we design antibodies against the spike protein, so these antibodies will go and bind to the spike protein before the virus binds to the lung receptor. So this antibody S309 is against the spike protein. So they will go and neutralize this virus, because this protein is against spike protein, and the spike protein is essential to enter into the lung cells through ACE2 receptors. And then virus is totally covered by the antibody, it cannot enter. This is also the principle for vaccines. So we take mRNA, right? We take mRNA for spike protein. So our body, after 8 to 10 hours or 24 hours or 48 hours, mRNA produces spike protein. Then our body produces this antibody because spike protein is foreign to us. And then we go to mall, anywhere we don't know, we get virus, but we have this antibodies from vaccines. Plus, at this stage, we also have memory cells. So we get virus again, the antibodies are ready to take over the virus and would not let the virus enter into the lung cells through ACE2 receptor. So what did we discuss in the class? This was a simple class, simple chapter to talk about monoclonal antibodies, how monoclonal antibodies can be used as a treatment. So treatment. In next class, which is on, this is Thursday, right? So on Tuesday, I will teach a little bit about how antibodies can be used for diagnosis. And then we will finish this chapter. Any questions?
No, Professor, no question.
Okay. So anybody wants to say that who came later on? I can mark.
Bernard.
Few I remember.
Bernard Sajin. BRUNARD. That's the
Yeah,
last
I
name.
saw you.
Thank you.
Anderson Hoya. Bajwa, you are here. Basu Masata, are you
Kia
here?
Melvin.
Basu Masata? Basu Isata? Donald Kiara? Lakisha? King McFarlane Dakres? Okay, so I think anybody who came, I have marked him or her present.
Oh, Anika Thomas.
Yes, Anika, I got you.
Okay.
Kia Melvin.
Yes, I saw you.
Okay, thank you.
So if there are no questions, then I end this.
Professor
Yes.
Shamika Lawrence.
Yeah, Shamika, I saw you. Yeah.
Okay, all right, thank you. Bye.
Okay, so any people who have their midterm, good luck and see you Tuesday.
All right. Thank you, Professor. Have a good evening.
Good evening, bye.